Well, good afternoon, everyone. Welcome to Northeastern at noon on this bright and sunny day, very appropriate for Earth Day. My name is Joe Hunt, Engineering 62. And I mentioned Earth Day, and it's very nice we have a topic that fits very well with Earth Day. And that's a presentation that will give a brief overview of all the many projects that are going on at Northeastern's Marine Science Lab. And in addition, will give us a focus on a project called the Mission 31 program. Now, the Mission 31 program, which took place in the Aquarius lab, which is the only underwater lab and habitat in the world. It's located about 9 miles off Key Largo and is managed by Florida International University. Now, all of the presenters at one time or another have participated in projects involving the Aquarius lab. The principal host for today from the Marine Science Lab at Northeastern is Professor Brian Helmuth. He received his bachelor's degree from Cornell University and a master's from Northeastern University, both in biology. He went on to get a doctorate from the University of Washington on zoology, and then moved on to do postdoctoral work at Stanford University. Then moved on again and became a professor at the University of South Carolina. Today, he's here at Northeastern as a professor who has a joint assignment, which means he's associated with the Department of Marine and Environmental Sciences, as well as the School of Public Policy and Urban Affairs. Now, his research interests focused currently on the effects of climate and climate change on marine life using a variety of techniques, including fieldwork, remote sensing, as well as mathematical models. Now, while much of his work has been focused on coastal regions in North America, he has also contributed and collaborated with research projects in Australia, Chile, China, Hong Kong, Italy, South Africa, and the United Kingdom. Now, his work to date in this project seems to indicate that coastal environments are much more complicated than we've thought previously. And as a result, we may see effects of climate change on areas that we least suspected. For example, northern regions warming up first versus southern, more warmer climates. He's authored a host of publications, almost too many to count, or co-authored them. One of the interesting things about these publications that were made for peer-reviewed professional journals is that they have been cited by many other authors. In particular, he has over a dozen papers which have been cited by over 100 other authors, some as many as 300. And I mention that because it's very good gauge as to how important one's research is when it's cited by others in the field. I mentioned the Aquarius Lab project, and without further ado, I will turn it over to the host, Professor Brian Helmuth, who will introduce to you other members of a team that participated in the Mission 31 project. Professor Brian Helmuth.
Thank you. Thank you for that introduction. I won't go through all the bios of my teammates, but before I start, I want to introduce our team who will be speaking to you today. We have Liz McGee, who is the coordinator for the 3Cs Marine Biology program at Northeastern, as well as our dive safety officer. Sarah Williams, who is a technician right now, but is about to become a graduate student. Francis Choi, who works in my lab as my lab manager. And Dr. Mark Patterson, who joined me at Northeastern 2 years ago and is a professor in marine environmental sciences and the College of Engineering. We were all brought here as part of the Urban Coastal Sustainability Initiative, and hopefully at some point you've heard about that initiative. It was launched in the School of Science, the College of Science, by Dean Murray Gibson and our director, Jeff Trussell. And it focuses on a lot of the things that Joe was talking about. We really are at ground zero here in the Gulf of Maine. The Gulf of Maine is warming faster than 99% of other large bodies of water on Earth. We're already seeing very large impacts of climate change. 2012, for example, we saw a record harvest of lobsters. A lot of that is probably due to the increases in temperature. What we want to tell you about today, though, is an adventure that we got to participate in with the grandson of Jacques Cousteau last year. And the reason we did this is because it's kind of the ultimate example of how we can live sustainably on coastlines. So we thought this is an appropriate topic to talk about today with Earth Day. So what I want to do first, though, is take you back in time just a little bit, about 50 years ago, where some of us were either growing up or had kids growing up. And this hopefully should be familiar theme music to a lot of you. Does anybody recognize this? Yeah. This is why people like Mark and I got into marine biology. This is what excited us. This is really what has gotten us out of bed in the morning. And this is Jacques Cousteau. Jacques Cousteau invented scuba diving. He took the world underwater for the first time. He got generations of people excited about the idea of working and living under the water. That vision kept growing and growing throughout the 50s and 60s into the 70s, and evolved into this idea that someday soon, we were going to live underwater. We're going to live in underwater habitats. We're going to have underwater cities. And that vision is really what launched decades of research looking into whether or not we can live underwater. The first experiment was the Conshelf mission, which took place a little over 50 years ago, where a team of aquanauts lived underwater for 30 days. Jacques Cousteau oversaw the science team from the surface and dove down and lived underwater with them as well. And this is really what kicked it off. I'm just going to play just a couple minutes of video from World Without Sun. Yeah, if we could, please. I don't.
Where the sun never shines.
I think I have control. Thank you. So this is what I grew up watching. I mean, this is what really got me going. And so I learned to scuba dive when I was 16.
Is shaped like a disc.
Let me ask,
to avoid
how many people
snags.
Here have dove in the past or actively dive now?
Water jets that swivel.
So what I want to do is I want to
place
just really
of
quickly
propeller
walk through
and rudder.
what the limitations are of diving. Why was this so difficult? Why was it? Why did it really take a visionary like Jacques Cousteau to get us underwater in the first place? And it really has to do with the physics of how our bodies take up different gases. Ooh. This is what it's like at about 90 feet, all right, 30 feet. Thanks for the demo. In a nutshell, what happens when we dive is because we're at increased pressure, our bodies take up those gases at that increased pressure, and so we absorb more of that, those gases. Things like oxygen we use up really rapidly in our bodies, but there are things called inert gases, gases that our bodies can't metabolize, we can't do anything with, that stick around. And they tend to build up in our tissues, and nitrogen is the most infamous of these things. So if anybody's heard of the bends, what that occurs is when we go underwater for a long enough time that our bodies take up that much nitrogen, then if we don't come up soon enough or we don't come up slowly enough, those nitrogen molecules form bubbles, and they can lodge in things like your brain, they can lodge in your lungs, they can lodge in your joints. This is first noticed with caisson workers because they would come up after being in a pressurized environment bent over because of the bubbles in their joints. So the way we get around this, and this is part of what Cousteau and then a legion of researchers after, mostly with the Navy, learned, is that we can limit our time underwater. So for example, if we go to 60 feet, we have about an hour. If we go to 90 feet, we can say about half an hour. And the amount of time you can stay as you go down decreases with increasing depth. And so really bad things happen if you come up too quickly. If anybody's ever seen one of these videos of what happens if you drop a Mentos in a bottle of Coke, or even if you just shake a bottle of Coke and you open it up, it's the same thing. You're basically taking something that's in solution, you're agitating it, and you're making it come out of solution violently. So when you're opening up that can of Coke, you're opening up the pressure and you're releasing it. aspect, I did one of these saturation missions that I'll tell you about in a minute, where we had someone who was sick. They took a blood sample from them, took it to the surface, and it bubbled and blew the lid off of the container because there were so many bubbles coming out of that. So the way around that then is called saturation diving. And what we do then is we allow our bodies to completely saturate with that nitrogen. We let it take as much as it can so that the amount of nitrogen in our bodies is equal to that of the outside environment. So we're at the same pressure as the outside environment. So as long as we don't go any shallower than what we call hatch depth, any shallower than where our bodies have become equilibrated, we're fine. And so that means we can spend almost unlimited time on the bottom. Liz, in fact, broke some records when she was living underwater. We can spend 8, in Liz's case, what, 16? No, how long were you out in the reef at a time? What? Up to 9 hours. So normally if we have 1 hour, then we have to come up and we call off gas. We can then go back down for another short amount of time, but we can spend almost unlimited amount of times on the bottom of the ocean. We can do very delicate tasks. We can do observations that otherwise would have become impossible. So this led to a series of experiments that started with the conch shell missions, most famously the Sea Lab program with the US Navy. That ended tragically with a death that still is unsolved, what happened, but that really put a damper on underwater saturation. And along with that, kind of this vision of moving forward, of living under the ocean. So in a lot of ways, the same thing as the manned space program now. We had this excitement in the public. We had kids who really wanted to do this. We had awareness of the ocean. And that kind of dissipated. It kind of went away. So a big part of what we're going to tell you about, and we'll hear about some of the outreach we did, we'll hear about what it's like to live underwater, is trying to rekindle that excitement, trying to get people interested in the oceans again, with the plight of the oceans, but also how we can solve some of these problems. And as Joe mentioned, we did this using the Aquarius underwater habitat, which currently is the only underwater habitat in the world devoted to science. So I'm going to pass this over to my colleague Mark Patterson, who's going to tell you he's been more involved in Aquarius than anyone else I know, and we can go from there.
Thanks, Brian, and thanks, Joe. It's great to be here. I've talked to a few of you before this started, and I see we have a lot of electrical engineers in the audience, and I just wanted to issue an invitation to those of you who are interested in underwater robots to come out and visit my laboratory at the Marine Science Center. I'm a double E self-taught, and I've got a robot fleet out at the Marine Lab. And I'm very partial to the potential for robotics to expand our understanding of the ocean. However, I also believe very firmly that putting people into an environment is the best way to do the best science. So I'm going to keep my remarks brief because I want you to hear from our own scientist and 3Cs coordinator Liz, who actually got the privilege of living underwater in this unique mission that we helped Fabien Cousteau with. I've spent 89 days of my life living in habitats like Aquarius. Not all at once. The longest was 16 days at a crack, although I happily would have spent a month on the seafloor. The reason I enjoy going into this inner space station is that I actually take the laboratory down to the seafloor. This is not just a place to go to sleep at night and have hot meals and relax after a long day in the water column that you'll hear about from Liz McGee in a second. It's got power. It's got connectivity to the internet, so we can do some amazing science that we couldn't do any other way. So when Fabien Cousteau approached us about participating as science advisors for the mission because of our prior experience, we were essentially all in. So he was the mission leader. Fabien went to Boston University. And by his own admission, he wasn't the best student. He's definitely one who extols the principle of Northeastern's educational system. He learns best by doing, experiential education. He spent his life, like the Cousteau family, trying to bring issues that are important to ocean conservation to the consciousness of the public. And so we hosted him here with the help of Stanley and Jan Burba, a family in Salem, during November 2013, and then we participated as science advisors during the mission. As Brian mentioned, this mission in part was to show the ultimate example of trying to live in harmony with an extreme environment, that is the bottom of the ocean. There's our crew on the back of the support vessel that we went out every day to support this mission. So while Liz was inside living underwater, we had a team of graduate students and 2 faculty members topside. And we could also act as a conveyance vehicle to bring the numerous press and dignitaries who want to come out and experience vicariously what was happening on the bottom of the sea. We did do a lot of science, and you'll hear about some of the science later from Francis and Sarah, as well as Liz. But a very important theme of this mission, of course, given that we had a Cousteau in saturation, was reaching the public. So we used the bully pulpit of Aquarius because it's hardwired to the internet over a radio link from 9 miles offshore to try and reach the world. And Brian and I both saw this as a fantastic opportunity to help raise awareness of Northeastern as a world-class premier university, and also the Urban Coastal Sustainability Initiative that is front burner. I'm going to show you a little clip here that NOVA did on the mission, and I have to give Liz lots of props for being a very effective communicator. You'll see her in a couple of clips here talking about what's going on in the habitat. Having someone who's likable and can distill the science down into something that makes you really interested is a key part of science communication, and she did an awesome job doing that. So let me run this little thing, and I may hold the microphone up to the speakers so you can hear the soundtrack. Oops. Do I have to click on it, Francis? There we go.
Somebody today asked me if I miss like running around in a field or something. I was like, no, I'm running around on the bottom of the ocean.
For the last 2 weeks, I've been living underwater as part of Mission 31. I was a mission scientist.
You're here! Welcome to your new home.
Thank you!
Good to see you. Are you ready for this?
Aquarius is an underwater habitat. It is on the sea floor at 63 feet, and it houses up to 6 people at a time so that you can conduct underwater research. It's a one-of-a-kind space underwater. There's no other marine lab underwater in the world. The habitat is small, it's cozy, it's about the size of a school bus, 400 square feet.
And as you can see right outside the window, there's the ocean floor about 63 feet down. There's some fish swimming around.
Normally when I'm a surface diver, I'm always worried about my bottom time. When you're diving to about 60 feet, you have about 60 minutes of bottom time without risking the bends or decompression sickness. And that's not very much if you're setting up experiments and trying to do science. But as an aquanaut, as a saturated diver, because we are living underwater for so long, we can dive for up to 9 hours a day. For our 2 weeks underwater, we accomplished what we could have done in about 2 years from the surface. We came with 5 different science goals, overall looking at global climate change and how it might affect a coral reef such as Conch Reef, where Aquarius is at. And so we took the approach of looking at tiny microclimates such as inside a barrel sponge or inside the mouth of a tiny coral polyp to address what might happen if we have warming of the oceans or if they're becoming more acidic in the environment that is surrounding these organisms.
Like chin strap on?
I think one of the most intense moments I've had underwater was during this first dive with the helmet, where we had to flood the mask. We had to flood the helmet by laying down and breaking the seal at our neck and letting water in, and then we had to stand up and clear it out. And it was an instantaneous thing. It took less than 15 seconds, but I was still pretty nervous about that. After all of the diving I've done, that is one of the most scary moments. Sylvia is a Goliath grouper. She hangs out around Aquarius habitat. They are able to use their anatomy to create this sound wave that travels through the water, which is their way of stunning prey or communicating with each other. There was a moment where I crept towards her and was actually able to just hang out with Sylvia for about 20 minutes. And then right before she left, she boomed right at me and it went right through my chest. It's like being in a big rock concert or something. And then she just kind of went away. Like, all right, enough's enough. You had 20 minutes with me. It looks like an aquarium from your viewpoint, but you are the aquarium. It is the ocean, and fish are looking in at you. And at night especially, it starts out as tiny plankton swarming to the light around the habitat, followed by larger types of planktonic animals eating those, and then small fish, and then bigger fish. It was like the food chain unraveling in front of the viewport every night. It was awesome.
All right, we have permission. Secure your exhaust valves. All right, commence blowdown. Here we go.
It was very regimented, and okay, put your fins on, get your mask on, get in the water.
All right, passing 40 feet, 40 feet.
I didn't get to reflect on it until we were hanging on to the line, and they were telling us to come up to the surface one at a time. And we're all hanging on the line, and I'm realizing we spent 2 weeks underwater, and this is it. I got kind of teary-eyed at that moment while hanging on to a line at 15 feet. I didn't want it to end because it was so cool, and it was probably one of the most unique things I'm ever going to do.
GBH put together with our help and the Mission 31 team, and Liz made several great points that she'll amplify on here in a second. It really is one of the coolest things you can do as a scientist. NASA rents this facility periodically for its astronauts for training, and I've talked to astronauts that have been up on the space shuttle or the ISS and have also done a mission in Aquarius, and they say, you know, it's really a hard call. A zero-G is cool, but I threw up a lot, and Aquarius had just so much life around it. It really was neat to live there. So we feel almost as privileged as astronauts by what we've been able to do. The experiments that you'll hear about on the science couldn't be possible in the laboratory. Corals, sponges, and their relatives, if you collect them and try and take them back to a laboratory, even one with running seawater on shore, it's not the same environment. They will not behave the same way. We do experiments on the seafloor next to the habitat. We put corals into, say, experimental chambers, and the coral doesn't know that it's left the reef because it hasn't. So that's the real value of this underwater space station for our research. You already learned some of the facts about the habitat. It's an amazing piece of engineering. It weighs 81 tons, and it's 63 feet deep. It's about 40-odd feet long, 10 feet in diameter. But it sits on this giant leveling platform called the quadrupod. And it was designed so that if a hurricane was going to make a bullseye hit on the habitat, that they could raise the habitat quickly along with the quadrupod, put it on a barge, and get out of Dodge. But in practice, they don't do that. They actually had some close calls where hurricanes have brushed by the habitat, and the habitat has shifted a little bit. It starts listing a little. They've had some minor leaks, but then the amazing support crew comes out and pumps the water out and levels it, and they're back to business. So Mission 31 was 31 days long, 1 day longer than the Conch Shelf 2 expedition in the Red Sea, which was Fabien Cousteau's grandfather's mission that he was honoring. It's the longest mission in the habitat's history by, I don't know how many days, at least 10 or 15 days. 19 days was the longest before that, says Liz. And this was the 50th anniversary. That's why Fabien wanted to do this in 2014. I mentioned before the unprecedented opportunity to get the good news out about Northeastern and what we're doing on the environmental scene of the planet. The media team did an analysis of the outreach of how many people planet-wide heard about Fabien Cousteau and Mission 31, and then, thus, of course, a lot of them would hear about Northeastern's involvement, and it was greater than 500 million people planet-wide. So this is probably the biggest branding event in the history of Northeastern, which made folks on Columbus Avenue very happy. But we did not sugarcoat it. We did talk about the serious problems facing the planet. The oceans are under assault from a whole host of issues, largely due to what we're doing to the planet, and we didn't shy away from getting that message out and what could be done to address it. We had lots of help largely due to connections that, of course, Fabien Cousteau had. Sir Richard Branson Skyped in one day. We had several Hollywood actors that the younger generation knows. One of them's from Entourage, the other one's from the show Vampire Diaries. If you're going, oh, who are these people? I don't recognize them. That's okay. You're in the same demographic as me. But it was important to engage with a younger audience as well as an older audience on communicating this. So we had lots of luminaries show up. My favorite luminary in that bunch there is the woman that I lived myself for a week with 2 years before. That's Sylvia Earle on the left there, who is known as her deepness by the New York Times. She's been declared a national treasure by the Library of Congress. She was the first woman to lead an all-female saturation team in the Tektite habitat back in 1970, which earned her a ticker tape parade down Broadway and a dinner in the White House with President Nixon. And she's a force in nature. She's almost 80 and is still going strong, spreading the word about saving the oceans. She was the first chief scientist at the National Oceanic and Atmospheric Administration, by the way. I'm going to show one more quick video here about Northeastern's involvement, amplifying a little bit on the science, and then I will pass the microphone to Liz McGee, who will tell you about the experience of actually living there. And if you got any burning questions, especially when Liz is talking, we don't have to hold them to the end, because sometimes something really bizarre will come up with what she's about to say, and you'll go, what? How does that work? So let me play this little video here, Brian, if you could. Oops, sorry. A little bit. Here we go.
Somebody today asked me if I miss like running around in a field or something. I was like, no, I'm running around on the bottom of the ocean.
Is this the same one? Oh, we had a little media glitch here. This sometimes happens when you have high-value production like we do. So that's the same video as before. So why don't I pass it to Liz and let Liz take over, okay? So here's Liz McGee.
All right. Thanks, Mark. And I do really owe it to Mark and Brian for this opportunity to live underwater. And as I said in my video, it was, I hope not a once in a lifetime, but certainly an adventure of a lifetime. So while I'm talking, I'm going to be playing this video. Here we go, down here. Hopefully you guys can see it. This is a video from a camera called the Edgertronic camera that we got to play around with for hours underwater. And I mean hours. One shot took 3 hours to shoot. We would set up the camera and get it perfectly weighted underwater, and we would be kind of on our heads because the helmets are heavy, looking through the camera's viewfinder at whatever creature we were trying to film, these tiny little creatures, and we would wait for hours for the shot to happen. And that really is not possible to do while doing normal scuba diving. It was a treat for me. I've done quite a bit of diving in my scientific diving career, and this was the most unique experience. I was doing a lot of diving, about 9 hours a day, in this helmet that you see me in this picture. It was about 40 pounds, and it took about 10 minutes of setup and testing to make sure it was secure. And upon entering the water, I could actually communicate with the Aquarius habitat and reef base 9 miles onshore and my buddies. And so this was the first time in my life that I was ever able to communicate, verbally at least, while diving. And I was frequently told to please stop talking because I was talking so much while underwater. I couldn't contain my enthusiasm, and I still can't contain it when I'm talking about the mission. It was just really that much fun. We also did some other really great science that we're going to talk about in a moment. But up next we have... This is the video we were looking for, right? No? Oh yes, this is our...
Been wondering what we've been doing while we've been spending hours in the water. And the thing that's been taking most of our time in the water is the plankton trap setup. So we set up these plankton traps in the afternoon, and then we collect them in the morning. And each one takes about 5 minutes to set up, and then we deploy them on 3 different substrates: coral and sponge, live rock, which means it's encrusting with anything but coral and sponge, and sand. And then in the morning, we go and we harvest the traps and cap them quickly to trap all of the plankton that has gotten in them overnight. And we send them up with the surface team where they put them in ethanol, and then they're going to analyze all the different species of plankton that are in them.
So we get the fun of actually sorting through tens of thousands of individual organisms ashore. And right now we're doing it with a time-honored instrument. It's called a graduate student with a microscope. Looking at the plankton is one of my favorite things to do, and there's so many different phyla. These are major divisions of life on this planet that are swimming around at night, reproducing, feeding the corals. It's been found that the corals themselves, they need those plankton for an energy input at night, and if they don't get enough of it, and there's a stressful event like a warm temperature event that can lead to coral bleaching, if they don't get enough zooplankton, they actually won't make it through that event. So that's why we're monitoring the zooplankton here, which has been wonderful.
So I joined the mission halfway through and switched out with 2 other Aquarius scientists. And I went down with my fellow Aquanaut scientist, Grace Young, who is now at Oxford studying ocean engineering. And we spent 2 weeks training for this mission. It was pretty intense, but really exciting. And then we were able to saturate. Once we got down to saturate, we would wake up every day and have a jam-packed day, usually starting at about 6 a.m. with a 3-hour mission where we would go outside, collect plankton, for example, do some other tests, come back in, Skype with about maybe a dozen classrooms within a day where we would actually sit and have wireless internet while underwater and communicate with classrooms around the world about what we were doing. And then we would go out for a midday mission, another few hours. And that one was usually not on the helmet, but probably just regular scuba diving, where we have our tanks on our back and we can just go. And then we'd come back in, definitely eat a bunch and drink a lot of hot chocolate to warm up, because we were burning a lot of calories while out in that water. Although it was 85 degrees, you do burn a lot of calories to keep your body temperature up. And then we'd go back out for another evening mission, about 3 hours again, and come back in, and we'd spend about a half an hour talking to the surface team, closely coordinating what we were going to be doing for the next day. So I would communicate to them, I'm going to leave the plankton containers under the port side of the habitat. And hopefully I'd remember that the next day, because they only have 60 minutes to come down and collect what I've collected for them to bring it back up to the surface. So each day was long and exhausting, but totally exhilarating. And it was with close choreography with the surface team that we were able to make it happen. So now I'm going to bring it over to Sarah, who's going to introduce all of the science that we did.
All right. Hi, everybody. So like Liz said, we spent a lot of time Skyping with her. We had a lot of stuff to do on the surface when we weren't diving. We got about 3 dives a day in, but we had to go through and sort through samples, get all of our gear prepped, and figure out data analysis, figure out our outreach schedule, all on the surface. So we had a lot of late nights. We were up until midnight, 1 a.m. a lot of the time, just getting everything together for the next day. Let's see. So up there you have us Skyping with Liz. It would usually just be FaceTime on one of our cell phones. This video is just our boat ride out to the site. We had about a 20-30 minute ride out to the site every day. And that time we weren't just hanging out on the boat, even though that's what the other people at Aquarius thought we were doing. We're normally just getting samples ready, getting our equipment ready to go, because we only had a limited time at the Aquarius site to get everything done. And we had a lot of stuff to do. We had, I think, 4 different projects going on. You had the zooplankton that Liz was talking about, and then we had sponge science, if this will move along. So we had zooplankton that we were studying. We were studying sponges. We were studying corals. We also had some samplers out on the reef to look at environmental contaminants. But I won't get into that, but I will start talking about some sponges. So sponges out on a coral reef don't look like your kitchen sponge. And a lot of the time they look sort of spongy still, but we're looking at the giant barrel sponge out on the reef, and they're often called the redwoods of the sea because they live for a really, really long time. And they are very important for coral reefs because they filter the water over the coral reef in 2 days. So all the sponges on that section of the reef are filtering the water, and they get through that entire fluid volume within 2 days. So really important for understanding what's going on a coral reef. And we were able to study these guys by deploying just a suite of environmental sensors inside of sponges using tripods. So we would just bring the tripods down with us, usually coordinating with the Aquanauts because they would do a lot of the setup time for us, but we brought the instruments down to them. So we would bring, let's see, go back. So the one in the middle is a sonde, an XO2 sonde, which is basically a bunch of sensors combined into one. We were looking at dissolved oxygen. We were looking at chlorophyll, kind of what plankton's in the water, and also looking at salinity and pH inside the sponge. And then the big white sensor inside the sponge looked at the water flow within the sponge to see how fast that sponge was pumping water throughout it. So the graduate student in charge of the sponge science was Allison Matzel, and she's using this work for her PhD. But now I'm going to talk about corals because that's what I'm more familiar with. So I am really passionate about understanding corals, and specifically the individual units that make up coral colonies called polyps. So the corals of the coral reef are actually colonies of these individual units called polyps. And these polyps are connected by their internal plumbing. They have all these canals that connect each polyp to each other, and they're able to share resources that way and work together as a whole organism. So I like to say that I'm kind of like a coral brain surgeon because I use these really tiny microsensors, which are made out of glass and they're thinner than your hair, and I insert these directly into a coral polyp's mouth to measure what's going on inside that individual polyp. And I'm measuring oxygen most of the time because that's a very important metabolite for what's going on in the coral system itself. So let me play this video. Let's see. So during Mission 31, we were able to set up our Unisense microsensor system inside of corals for 24 hours at a time to get a really good idea of what's going on with the oxygen dynamics inside of these coral polyps over a very long period of time that hasn't been, an experimental time that hasn't been measured before because this is the first time that the system had been deployed in the field. And so that's me inserting our sensors into corals right on the platform of the Aquarius habitat. And since I only had a couple hours at a time during the day, I would set it up and the aquanauts would keep an eye on the sensors, make sure no one's knocking them over and that no fish does knock it over, which did happen. The Goliath grouper did knock over my microsensors once at least and broke them. So we're working in pretty difficult conditions. And since the microsensors are so small, I have to wear this very special headgear over my mask, and I look like a big nerd. And they're just like jeweler's headgear to magnify what I can see. So I would set that up using a very fine movement micromanipulator to work the microsensor into the coral polyp's mouth. And when our measurements are then matched with all the environmental data that we were looking at over the reef at the same time to figure out how corals are dealing with that environmental stress, changes in temperature, changes in flow, and seeing how that changes how they're using their resources within a colony. We also had a lot of fun. So we were out on the boat together all day for about 2 weeks, and there was quite a bit of pranking going on. So one of Mark and Brian's favorite pranks from their time during missions was filling your booties, which are your basically boots you put on into your fins to keep your heels from rubbing. People would put dye inside of the booties to dye their feet. So you can see we pranked Mark, his feet are purple. So we had a lot of fun. It wasn't just science, but science is fun is what we were also trying to communicate. And so talking about communications, I'll turn this over to Francis Choi.
Well, thank you. So, so I just want to mention that all the science you saw with the corals, Sarah's going to be continuing doing that because she just got into graduate school with Mark Patterson for the next 8 years. Maybe 5 years. So she's going to continue working on the microsensors and continue to do gut surgery on poor corals around the world. But I wanted to talk to you guys about what we did. Mark mentioned reaching out to 500 million people around the world, and so I want to talk to you about how we communicate it as the Northeastern group to the general public. And so we wanted to start out doing, well, social media is one of the main things that we got in. So within that 500 million outreach, Facebook was involved with Northeastern, Twitter, Instagram. We try to hit up all the social media that, you know, the 20-odds or the teens will be interested in, and check out videos. And with live Wi-Fi down in Aquarius, we were able to give out real-time live experience out to the world as it was happening. So if we saw a big fish, we would, you know, tweet about it or Instagram about it. And one of the main vessel of communication as well is with the Museum of Science in Boston. Our lab and Mark Patterson's lab, we have a really good relationship with the people at Museum of Science in Boston. So we had 4 events going on during Mission 31, and each time the surface team got to go inside Aquarius and become kind of like the liaison of Boston back to the people in Boston. So we got to answer questions. We got really quite intimate time with our audience out in Boston. And at the same time, we had scientists as well. Here we had Dr. Fernandez showing us about her cutting edge science that's going on in Northeastern, marine science that's going on as well. And so we had a couple of events out in Museum Science and showing off what the science is going on and how living under Aquarius is as well. And so since Mission 31 has ended, we also think about, well, where do we go from here, you know? We don't want this to be a fad. We want this to continuation, you know, the exploration, the research, and also just talking about, you know, Earth Day and science as well, right? How to protect the ocean. And so our lab we purchased this virtual tour, this 360 Hero machine. And what it does is it makes virtual images or virtual videos of the surrounding you put it in. And because it's underwater, we're able to really capture what it's like to be diving underwater. And I would like to show you guys one of these videos really quickly. And we're really able to capture what it's like diving underwater and what it's like living in Aquarius. And so let me just open up this fancy program here. And this will take no time, I promise. And there's audio to it, but we're just going to show you this. I'm going to talk over. And so not only can you see, you know, the spotted eagle ray flying around, you can really turn around and immerse yourself in this environment. And so it's a good way to reach out to right now the upcoming generation that likes to use their iPads a lot, the phones a lot as well. But because we had such a good relationship with Museum of Science, we decided with this, we worked with them, with the planetarium, to continue to make, you know, public outreach videos that involve these kind of things, so we can able to project these 360 videos at the planetarium in the future to talk about ocean exploration and how climate change are affected by the warming of the oceans or changes in the oceans as well. So with that, I would like to finish off our presentation and thank all of you guys for coming out today for the noon talk. So thank you, and I'll end with this time-lapse video of Mission Aquarius.
So there were 6 people living on the bottom at all times. We divided the saturation team into 2 groups, as Liz mentioned. So Liz and Grace were swapped out with 2 other scientists from Florida International University at the halfway point. And then Mark and I led the surface mission team. And so we were the ones who were going back and forth, living on shore, coming out in the boats, and we were restricted to surface diving. But we had tight coordination with Liz and her team on the bottom. No worries.
It took 17 hours of decompression for us to be able to come up. And so we weren't doing anything besides sleeping, actually. I was sleeping through it. But there was somebody, a technician, that came down to decompress us. And he was in charge of slowly bringing the pressure inside the habitat to what it is on the surface. So the habitat stays where it is, but it's a self-contained unit that they can control the pressure of. And so over those 17 hours, they slowly brought us up to surface pressure. And then once we were there for a certain amount of time, they blasted us back down, opened up the hatch door, and told us to get out. And so we got our scuba gear on and swam up. And we didn't even have to do your standard 3-minute safety stop, which you normally have to do for a scuba dive, because we were already desaturated, basically. So it was pretty cool. Yeah. Yes. Getting in and out of the habitat, yes. Yes, so it's equal to the pressure at about 45 feet where the habitat sits. And there's a room called the wet porch that is separate from the living quarters of the habitat. And it is not air conditioned, and the pressure is kept equal basically to that of the water right outside. It's slightly positive, kind of like when you turn a cup over in your sink, and it's got a pocket of air. That pocket of air doesn't really go anywhere. The same kind of theory is working with the wet porch, and you just step down steps and duck under, and you're into the water. It's pretty magnificent. Right, maintaining life in the habitat was a round-the-clock job, and there is I can speak to this in some detail, but Mark and Brian know a lot about it too. There's something called the life support buoy, which sits on top of the water. It's a big yellow buoy, and it's got a compressor and a generator, and that feeds the habitat with both compressed air for tanks, for filling scuba tanks, and breathing, and also power. That's how we got our power down there too. But also, we had I think we had over 30 surface divers helping us throughout the mission, and some of those were also U.S. Navy divers who were bringing down life support materials such as our water in gallon jugs, clean towels, which were vitally important, our food. Francis sent us pizza down one day, which was awesome to eat pizza underwater. And we had 2 habitat technicians that lived with us inside the habitat. They were the 2 of the 6 that were down there. And we also had somebody round the clock living or watching us at Reef Base, which is back on land. They're sitting there watching us on video with audio. If they need to, they can call us at a moment's notice. They also have a chat set up so we can communicate directly via the computers. So they were monitoring everything round the clock as well. Yep.
Several years ago, there was a terrible BP oil spill in the Gulf that devastated the whole Gulf. And the question I have for you is, well, lately they've been advertising on TV saying it's recovered a great deal. The question I have for you is, did you see any evidence of this spill? And do you have any comment on the recovery of the Gulf?
So a lot of what BP's putting out there is baloney about the recovery of the Gulf. One of the samplers that we put out, and these data are still being analyzed, so stay tuned. Loretta Fernandez is a colleague of mine in civil and environmental engineering, and we put out plastic strips around Aquarius specifically to look at whether there were still dispersants that were used in enormous volumes to disperse the oil from then it was 4 years ago, whether they were still emanating out of the Gulf. So her sensors should tell us whether there's still a contaminant issue from the dispersants, yet alone the oil. And those data still haven't been analyzed. But sadly, the Gulf oyster populations and the shrimp populations and a lot of the fish populations upon whose livelihood people's lives depend there has not recovered to the levels that we saw pre-spill.
Can I add something to that? So a lot of my work is funded by NASA, and I'm on listservs. I'm sorry? I'm sorry. Much of my work is funded by NASA, and so some of these listservs, we were watching the oil spill via satellite, and it came incredibly close to looping around the Florida Keys and moving all the way up the East Coast. At some points, we really thought it was going to make it. So it could have been just awful if that had just gone a little bit farther east and looped around. So the question then is, that was watching the main front from satellite, but how much of that was leaking? Were we really seeing the edge, or did that really make it around the corner? That's still an open question, I think.
So Northeastern, through a foundation I'm associated with, we have pending a proposal to get some of the BP money that Congress is awarding to municipalities all around the Gulf of Mexico. It's called the Restore Act. And Monroe County, which is where Aquarius is located, is eligible for those funds because it's got a Gulf of Mexico side as well as an Atlantic side, because the Keys are so thin and narrow. So we're hoping, fingers crossed, that this isn't the last time Northeastern scientists get to use Aquarius, that we will have 4, maybe 5 missions in the future if we get the award to us through the Aquarius Foundation. That's been pending for 2 years now. BP, as you know, has been fighting a lot of the settlements through the court system, and now the money is just starting to flow to help do some good in terms of job recovery and environmental education and environmental restoration.
Yeah, go ahead.
Similar to that gentleman's question, fire field or sea is what you want to say. You know the Malaysian airline that this way digging in the Indian Ocean. Is there any way the Navy people and Army people certainly know what they're doing, I hope. Is there any way your facility can aid in that, finding the difference in arms, if that airplane might have subject?
So I actually got a phone call from a ship on site. Yeah, so the question was about the Malaysian airline mystery and whether Northeastern is helping at all. So since I do robots, I actually did get a call from a person I know who was actually on the site doing the exploration, asking some advice. The robots that are made in my lab can't do the really deep water exploration that probably the debris field of the unfortunate flight is located in. But we can give advice. There's only a handful of institutions that do marine robotics, and we can all be proud that Northeastern is one of them now. We got not only my lab, but the lab of Joseph Ayers, who does robots that look like animals. He's made the robotic lobster and the robotic lamprey that some of you may have seen. And while they sound like curiosities, the robotic lobster was actually funded by the Department of Defense to help look for mines in shallow water. So there's a lot of overlap with the world of robotics and national defense when it comes to understanding what's going on in the ocean, including finding mysteries like where did that airplane go. But the ocean's a big place. Yes?
Could you contrast for me the way how this has been done before by, or the kind of missions that they've done on the Cape with the institution down there? But more importantly, you seem to have found a great way to live in an environment like this. Of course, you're on Earth, not up in space, but I would think some of this stuff where you've had lots of fun doing this is the way for future astronauts to go on missions to Mars or something. You found lots and lots of just ordinary activities that keep everybody happy.
Yep. That's a good point. Alan asked about whether, the fun factor and living in a confined space would be useful for going to Mars, and the answer is yes. NASA realizes the value of doing missions in Aquarius where the astronauts actually have stuff to do every day, like Liz had to do every day to advance a scientific goal, and that's what's going to happen with a long-duration manned space flight. So it's a great training facility. There's a man in the back there in the end.
learnings from the ethics?
So far from the science, is that what you're asking? So, okay, I'll give a couple and then I can pass it to Brian. So on the coral science side, we learned that the network that makes up a colony of those repeating units, the polyps that Sarah told you about, is actually quite complicated, and the behavior of the coral actually influences what's going on inside, in particular whether it's expanded or contracted. Corals are animals, and yet they move in slow motion a lot of the time, like it's almost like watching a plant grow or paint dry. You got to use time-lapse photography if you don't want to go crazy. I did want to mention, by the way, that we've done simulations of how the colony should behave using an electrical network model for all of those out in the audience who are double E's and love networks of resistors and capacitors and inductors. We actually have a electrical network model of how resources should be shared amongst the repeating units. So we got some very interesting dynamic data, again, wearing my double E hat, on the frequency response of how the colony behaves. Brian's student has been looking at the diet of the sponges. They eat things the size of bacteria, and she's found some interesting differences and some, again, behavior that these very primitive organisms are exhibiting. We don't know yet what the story is on the whether Corexit, the dispersant, is coming out of the Gulf still, because those samples are still being analyzed. And anything else I should say?
Actually, to me, the coolest part of this mission had nothing to do with the science, or very little to do with the science. I have two young daughters. The oldest is now 13, and she was able to go down on the mission with us. And so she did the surface support from the base, and she ran a blog. She advised us on what she thought kids would think was cool and what wasn't. And it was really exciting to see her get into it so much, and then just the impact of her seeing Liz and Grace underwater, and just that kind of aha moment that really, you know, there are no limitations to what she can do. To me, that was the best part of the whole mission. And I'll pass it over to Liz as well here.
I found out about the Aquarius habitat when I was Northeastern undergrad doing the 3Cs program in 2006. And I thought to myself, oh, that's cool, but I'm never ever going to get the chance to do that. I do some open water science diving, and that's about it. And then I found myself with this opportunity. And to me, that was the incredible moment, was being able to see where I've progressed in my career, and also talking to students of all ranges since the mission has ended. I think I enjoy that just as much as I enjoyed the mission, and I think from my videos you see that I really enjoyed it. So yeah.
Question for Sarah. Measuring dissolved oxygen is not all that simple, and particularly at a very high atmospheric pressure, which you would have down that deep. So I'm wondering how you calibrate your sensors.
All right, so great question. So the question was, how do we calibrate our dissolved oxygen sensors, because we're working at much higher pressures underwater. So all of the calibrations actually get done at the surface. And these probes work because they're measuring a change in partial pressure of dissolved oxygen. So at the very tip of the sensor is a thin membrane, and that oxygen diffuses across the membrane, and then we have a potential difference that's, it works, it's an amperometric sensor, so you're looking for a change in current within the sensor. But we do all the calibrations at the surface, just doing a 2-point calibration where we have fully saturated water with oxygen, and a anoxic solution. It's a linear relationship too, so it's just 2 points, and we just have to make sure that we know the salinity and the temperature of the water that we do our calibrations in, and as well as where we're measuring. It doesn't really depend on the pressure that we're doing the calibrations in. Yeah.
So in addition to that is the big YSI you saw earlier, the Exxon. We also have smaller pressure loggers out there as well, and salinity loggers as well. So not only we're doing, I guess for the micro sensors, they're calibrating on the surface, but for the larger equipment, we're calibrating at the site. So when we get all the data, not only we're collecting dissolved oxygen, we're collecting pressure, we're collecting temperature, we're collecting salinity, and so we're also collecting what's happening inside and outside the sponge as well. So when we bring all the data back up, we can use all of that different information to get a more exact value of what the DO is going to be down there. So.
I apologize. I actually have to run off to give another talk. So I'm going to leave these guys here, but just thank you very much, and I'm sure they'll be happy to continue answering questions. Thank you.
Okay. Up in the back?
It's probably unrelated, but I often wonder and worry about contaminated stormwater discharge into the ocean and treated sewage being discharged. What kind of an impact? People say, oh, don't worry about it, it's a big ocean, it can handle it. But are you seeing anything? Should I worry about it?
That's a great question. So yes, the ocean's been used as a dumping ground for raw sewage in Boston Harbor until fairly recently. Now we've got tertiary treated Deer Island sewage going out in 150, 180 feet of water through a big diffuser. And the question is, does that have any local impacts? We actually hosted the Massachusetts Water Resource Authority's annual oversight meeting at the Marine Lab this past fall. And our director, Jeff Trussell, was on that advisory board. And that was a day-long conference for the contractors that are hired by the state to monitor the health of the coastal ocean near that outfall to see whether there's an impact or not. And it was fascinating for me to listen to the presentations because I was learning about the outfall, and my robotics lab will actually start swimming robots out there this summer to look at the impact of not only the Deer Island outfall, which is 1/3 of a billion gallons of treated sewage every single day into the coastal ocean, but we'll also be looking at the ones near Lynn and the ones near Salem. The short answer is that tertiary treated sewage with proper microbial digestion, et cetera, and storage tanks for what's called combined sewer outfall runoff when we have heavy rains have greatly improved water quality near shore. In fact, the Inner Harbor is a success story of how you can clean up a place and make it somewhere that people want to recreate and go out on the water and catch striped bass instead of fleeing away from it like they did 30, 40 years ago when it was essentially an open sewer. That said, we have suspicions that this is still a big perturbation just because we have so many people with so much sewage being produced. To put it in perspective, nitrogen is often a limiting nutrient to fuel the growth of plants in the sea. And there was a contractor from Battelle who was studying the nitrogen budget and output from the Deer Island plant who got up and said, well, we've added it all up and made our measurements, and the Deer Island outfall is really only 5% influence. And I said 5% of what? It's 5% of the nitrogen input for the entire Gulf of Maine is coming out in one spot. So even though it's been treated, et cetera, it's still a perturbation. We're also worried about chlorine, which is supposed to be neutralized, but still is smellable by lobstermen and other people that make a living on the water when they go by that outfall under certain conditions. Some of the additives may have impacts on the very earliest stages of life in the sea, like the larvae of lobsters, for example. So it is a fact of life that a lot of coastal cities discharge into the ocean. They have it a little easier on the west coast where the continental shelf is not so long and broad, but you rapidly get into deep water close to shore so that you can use that aphorism that I don't like. The solution to pollution is dilution. We're hoping that part of our sustainability research at Northeastern actually is looking at different ways to handle sewage treatment. We've got several world-class experts in civil and environmental engineering that are helping to tackle that. I think the future, if I were to have a crystal ball and look 100 years from now, is that we won't be doing things at the mega scale necessarily. We might be doing things at the neighborhood or hyper-local scale for treating what we're doing so that neighborhoods or towns can have much more control instead of just hooking up their sewage to a giant mega authority. Alan's got another. or question.
So this would be a simple question. Maybe there's a simple answer though. As an old Vermont kid who grew up and worked on farms, we would of course have the manure and take it out, put on the fields, and then we would use lime to balance the acid, and of course our hay and all this stuff would thrive. Is there something comparable to that in the ocean? We do hear about the algal blooms, both red and the other kind that you get from runoff from agricultural stuff and so forth that cause problems with the algae. Is there some kind of organism out there that tends to thrive if it gets a lot of this human sewage, and there's some kind of thing that alkalizes it so that it becomes sort of balanced and can actually grow lots more algae as in the ocean?
So Alan asked a great question. Is there something we could do with, you know, pollution that has nitrogen in it from our activities on shore to somewhat neutralize its effect in the coastal ocean? And because of the volumes we're producing, there's no magic bullet. The nitrogen, it used to be thought that nitrogen was flowing into the coastal ocean largely from runoff from the land, but now we know better. We know a lot of nitrogen comes into the coastal ocean from cars in the form of NOx compounds that actually dissolve straight out of the air. And there are lots of organisms that love nitrogen in addition to the algae, which can sometimes bloom in great numbers and cause low oxygen areas in shallower systems like the Chesapeake Bay, which can be a big issue, or even the Gulf of Mexico, which gets an enormous amount of fertilizer out the mouth of the Mississippi. All plants love nitrogen. I mean, it's fertilizer. And one plant in particular that's very important in protecting our infrastructure near coastal cities are wetlands. And up and down the East Coast right now, the salt marshes, which consist of a handful of species, one of which in particular, the smooth cordgrass, Spartina alterniflora, they're falling apart because they're getting too much nitrogen coming in from the coastal ocean. They're actually growing top-heavy. They're getting too much of a good thing, and they're putting too much of their growth into the above-ground biomass and not enough into their roots. And now the marshes are sort of caving in on themselves during the growing season and eroding away. And this is really bad news because marshes are nurseries for economically important species. They do a lot of other ecosystem services like modify the water quality in the coastal zone. And they also protect against flooding when we get a big nor'easter or, you know, God forbid, a Hurricane Sandy hitting Boston in the future. You want your wetlands in good shape so that stuff along the shoreline doesn't get flooded. So there is some research going on trying to make essentially what you're after is, you know, is there some sort of additive or organism that before it goes into the sea could be added to do something like that? There are schemes for aquaculture in the coastal zone where right now aquaculture, if you grow fish, fertilizes the ocean and causes blooms and leads to dead zones underneath the fish pens. There are schemes in place where you would actually grow algae within the fish pens to suck up the nitrogen, and then you'd harvest the algae and sell it, you know, to the Japanese market where it'd be used for nori. So these sort of polyculture approaches are gaining some favor. And getting back to the hyperlocal angle, we had a job candidate visit from Florida last year who's actually designed a sewage treatment system that you install at the neighborhood level using plants that grow like in the median strips or even in little things just barely underground, so that as the sewage water runs off during heavy rain events from the streets, it flows through this biofilter, which is living, before it enters the sewage treatment system. And that's something that, you know, our scientists are playing in that space now. I think Joe wants to hook me and pull me off the stage, so.
Well, fortunately, if you haven't had a chance to ask a question, the team will be available for a short time afterwards. Feel free to come up here. I want to thank, as I think many of you do, the team here, and a special kudo to the impromptu light team who handled the lights during the presentation. We do have a small honorarium, if you could just pass these out. So once again, let's show our appreciation to Northeastern's Marine Science Lab. I have one quick announcement, and that is we have surveys available. If you haven't filled out a survey, I really encourage you to do so. We really covet those because Northeastern at Noon is not our program. It's not the university's program. It's your program. So if you have any thoughts or any ideas for future presentations, please let us know by these surveys or just talk to one of us on the committee. Well, I think it's probably time for us to go out and celebrate Earth Day by enjoying the great weather that the planet has provided for us. Bye now. See you next time.